Hey everyone, Professor Davis here, and welcome back to part 2 of Microbial World and You. So again, remember this is going to be an introduction to microbiology, and we are now going to focus in and dive into looking at the scientists who contributed to this field, okay, how it kind of came about. All right, that's going to be our focus here in part 2. So let's take a look at the history of microbiology, and our first player that we're going to look at is Robert Hooke. Robert Hooke started his kind of studies and with his findings in the mid 1660s. He was able to observe a cell for the first time underneath a microscope, and he's the one who actually coined the term cell. And this marks the beginning of what we are going to eventually see as the cell theory. Now, the cell theory doesn't come into full effect until 1869, and it has 3 components. The first component meaning all living things are made of cells, which Robert Hooke helped contribute towards. There are 2 other principles, though, to the cell theory that are important. So not only are all living things made of cells, the cell then is considered the basic structural and functional unit of life. So in order to be living, you have to have at least one cell. Also, life begets life, meaning cells are capable of reproduction. So these are the 3 things that make up the cell theory. They're the 3 kind of principles that make it up. Now, again, Robert Hooke helped contribute to the first one. Now, he called them cells because they looked like little boxes, which is what they called, where we use it for like jail cells or monastery cells, where they're these little boxes that people lived in. Now, he didn't have enough resolution to actually see most microbes with the microscope that he had at the time, but he was able to see those cells for the first time. So this brings us to another scientist who also developed a microscope, but his microscope was a little bit better. This guy is known as Leeuwenhoek. Leeuwenhoek did most of his work in the late 1670s. He is the one who developed a more advanced microscope that uses magnifying lenses, and he observed live microorganisms. So when we looked at Robert Hooke, he was looking at cork, which is dead plant. Here, we're actually seeing that he was looking at live organisms, and he used a lot of times pond water to look at this. And he saw that these little organisms were moving around because they were alive. So he called them animalcules. So kind of like animal molecules, because they were small and they were able to move. In reality, he was looking at bacteria and protists. Those were the things he was seeing. Now, he's best known for his work on the development and improvement of the microscope. And we see that not only the use of kind of pond water, he used rainwater, he looked at feces, he also looked at scrapings off of turtles. He looked at a lot of different things to help improve and use the microscope. The next contributor is Redi, and we want to look at him because he started looking into something that was a great debate during his time in the 1660s, which was spontaneous generation versus biogenesis. Now, spontaneous generation stated that things just spontaneously came about. So whenever there was rain, frogs just spontaneously came out of ponds eventually. We see that flies just spontaneously came out of rotting meat. But we now know that that's not true. Okay, biogenesis is the part that says that things come from other living things. All right, so there had to have been something present. Now, how did Redi disprove spontaneous generation and prove biogenesis? Well, he did an experiment right here where he used maggots in decaying meat. So the first container he had, he put the rotting meat in it. He allowed it to continue to rot, but it was wide open. So flies were able to land on it. He started noticing that these little structures called maggots started to form on it, and then more flies came out of that. Okay, so this is what he saw when it was open to the air. On the other side, we saw that he then took a container, and this was more of his control, where the meat was rotting, but he left a stopper on it where nothing could gain access to it. And so in this case, it showed that no maggots formed, no flies formed. And so this showed that the air and all that kind of stuff didn't actually contribute to formation of maggots and flies. So then he decided to do the same experiment with the meat, having the jar open to the air, but with gauze. So the flies couldn't land directly on the meat. However, they were still attracted to the meat. And he found that the maggots were developing on top of the gauze. And this is again where it's that relation, right? Eggs were laid, they hatched into maggots. Maggots then would turn into flies. That's ultimately what we now know, but this is what Reddy helped prove. And this shows that third part of the cell theory, which is life begats life. So life comes from other life. It doesn't just spontaneously happen, okay, like they thought here at this particular time. Another type of study to help support this spontaneous generation versus biogenesis is done by Virchow and Pasteur. So let's talk about them next, because they're in the 1800s. So we're skipping over a couple hundred years and we're going to Virchow in the 1850s. He was a major contributor to the cell theory of the life begets life, but he failed to present convincing evidence to the scientific community during his time. We now credit him with that finding, but back then they didn't really believe him when they looked at his work. We do see that Pasteur also helped with this in the 1860s, and he worked to support the philosophy of biogenesis by also creating pasteurization. Okay, so pasteurization is a big part of showing that life begets life, but it also helps keep our milk and different things like that microbe free for the most part. Okay, so microbes were in the air and they could contaminate a sterile solution. And so he developed this special kind of flask like you see here. When the flask was just straight open, air could get in and the microorganisms would grow in the actual flask. If we used this weird kind of shaped flask with the tube, the bacteria couldn't get all the way down into the actual substance, whatever was in there, whether it was milk or whatever substance was in there. So no bacterial growth happened. And so this is also what contributed to what we call aseptic techniques, which you're going to focus in on a lab where we talk about this, where we don't contaminate things. We want to make sure that we're only putting in there what we want in there and not contaminating it with something else. And so Pasteur helped with this. Now, because of this work in the 1800s, we see that this is known as the golden age of microbiology. So the golden age of microbiology runs from 1857 to about 1911. And this is when microbiology really exploded, where lots of scientists made contributions that helped in this field. Now, Pasteur is one we just talked about in the 1860s. He did a lot of good work, and part of this is that he helped develop what we call the process of pasteurization. And he did this because fermentation was super important at the time. Fermentation, guys, is the process used to make wine and beer. The problem is that bacteria was getting into some of the samples of the beer and wine, and instead of getting it to be converted into alcohol, it was being converted into vinegar. Okay, people don't just want to drink vinegar. And so because of this, it was hurting the industry. So Pasteur helped come up with a solution. His solution was what we call pasteurization. Pasteurization uses just enough heat. So it's the use of just enough heat to kill the harmful bacteria that leads to the spoilage, leads it to creating the vinegar, but still leaves behind the stuff we want, and it doesn't degrade the beer, the wine, the milk, whatever it is, in order to make sure that it stays, so it doesn't spoil as easily. Now, eventually, of course, some things will spoil like milk, but beer, especially wine, the longer it sits, a lot of times, the more rich the flavor is, as long as it's not contaminated. All right, and so pasteurization was what he contributed to this golden age. The next thing that the golden age of microbiology focused in on was called the germ theory of disease. So this germ theory of disease proposed something new. They proposed that diseases were linked to these microorganisms, these germs that you could not see. Now, this is not what they believed before. They actually believed before that diseases were due to they were punishments for your sin, for transgressions. But they are saying, no, it's not a punishment for your sins or your transgressions per se. It might be a consequence. But we see that they are caused by something that's actually present that we can diagnose, that we can see. Okay, this is what the germ theory of disease proposed. So some key factors or individuals who helped contribute to this is Semmelweis in the 1840s. He was looking at a certain disease that happens due to sepsis. They would call this childbirth fever. It happened after the child was born. And we saw that then the mom would get sick, have this fever, and then ultimately die. And this was looked at deeper by these physicians. And this is also known in our day and age called postpartum infection. But this was linked specifically at this time to physicians not washing their hands between patients, where they didn't wash it really well before they went to the next patient. Now we're thinking, oh my gosh, why would they do this? Back in this time, they didn't wash their hands like we wash our hands now. And even in 2020, we had to retrain people on how to wash their hands. So it's not as crazy as you think about, but this is a big thing that came about. Another individual who helped with this is Lister. Lister was in the 1860s. And he is the first one who used antiseptic during surgery. So he used a phenol product, which we'll learn a little more about phenol products later, but he used it to disinfect his surgical wounds. This actually helped reduce infections and the death rate drastically. So guys, when they were doing surgeries before, they wouldn't disinfect the skin before they started. Now we do this on the regular. If you go in for a surgery, they're going to give you an antibacterial soap to use. And then they're going to put that yellow kind of substance on you that's called like betadine or iodine on you and helps disinfect your body. Lister was the first physician to do this. And he used phenol. Phenol, though, smells really bad. And it can also irritate the skin. But we do see that it reduced infections and the death rate. And so again, making a huge contribution to the medical field here. So our next individual we want to look at is Koch. Koch is in the 1870s, and he came up with what we call his Koch postulates. Now, ultimately, what these postulates say is that he's able to link the cause of an infectious disease with a specific microorganism. So he was able to say that this disease that we saw right here is due to this particular bacteria. Okay, and he did this through an experiment with mice. And so he found a diseased mouse, one that died from a disease, and he took its blood and he looked for the suspected agent. Okay, so he took the blood, he looked for it, and then he cultured it onto an agar. Now, this bacteria grew, and so he collected it and he injected it into a healthy mouse. The mouse died. He then was able to then take that blood and culture it, and he got the same thing that was present before. Okay, so he was able to get the same causative agent. So he was able to show that what killed the first mouse ultimately killed the second mouse, and it was the exact same organism. Now, we do know that Koch's postulates are actually going to have some limitations to them because we do know a little bit more now about microbiology. We do see that some organisms cannot be grown out on a plate like you see here. Syphilis is an example of this. Some bacteria do not grow on agar in the way that others do. We also see there can be more than one type of organism that can cause the same type of signs and symptoms. One of the best examples of this is a UTI. A urinary tract infection can be caused by multiple types of bacteria or yeast. The most common is E. coli, but we would have to do an actual culture to find out what is the actual cause, because there's lots of bacteria that can ultimately cause UTIs. And then also one particular organism may cause multiple diseases. We see this with what we call gas. This is a streptococcus that actually can cause not only strep throat, but it can also cause different types of systemic issues, and it can also cause damage to the heart itself. And so because of this, it gives a wide range of signs and symptoms, but they're all caused ultimately by the same bacteria. So his findings are kind of limited now, but he made this huge advancement. Link a organism to a specific disease. The next guy we want to talk about is Jenner. Jenner was in the 1790s. In the 1790s, he was the first to create a vaccine. He was the vaccination that protects against, he came up with vaccinations that protects against certain diseases. What he determined is that these vaccines could help stimulate the immune system to provide artificial active immunity. Now, we'll talk more about what that means later, but artificial means it's been injected, but active means my body has actively made antibodies to protect itself. Okay, and so how did he do this? Well, he used cowpox virus, and he used it to help create a vaccine against smallpox. So he noticed that women who were milkmaids didn't get smallpox, but they did have these bumps on their hands from the cow, from the udders of the cow and milking the cow. And they called that cowpox. And so what he did is he scraped those blisters that they had on their hands, and he took that and made a solution out of it. He kind of purified it, made a solution, and then he injected his son. He then exposed his son to smallpox to see what would happen, but his son did not get smallpox. We find that his son actually made antibodies to help protect him against smallpox. So this is how the first vaccine kind of came about. Now, ethical? Probably not. But the whole point is that it did create a new branch that allows us to protect against certain diseases. Now, a key thing though, guys, is a vaccine has to allow you to make antibodies. If it doesn't allow you to make antibodies on your own actively and make memory cells, it's not considered a true vaccine. And there's a reason, because your memory cells are what allows to protect you long term. Okay? And so this is why we don't really call it a flu vaccine. It's called the flu shot. It only protects you for a short period of time, pretty much for a year. Then it could mutate and change. All right, but things like measles, mumps, polio, those vaccines have been shown to work for years and years and years, and they offer you pretty much lifelong protection. Every now and then you may need a booster, but for the most part, you've got those memory cells and it gives you a lifelong protection. So this brings us to kind of the last thing I want to hit on where we're talking about this intro to microbiology, and this is called chemotherapy. Now, when you think of chemotherapy, what comes to mind? Well, most often we link it to cancer, right? Chemotherapy is the type of treatment we use for cancer. But in reality, chemotherapy just means that we are using chemicals to treat a disease. All right, so yes, that is chemotherapy, but technically if you're taking any kind of medication to treat a disease, you are doing chemotherapy. Now, some of these are natural chemicals, and this is what we see with some of our antibiotics that we see, and some are synthetic drugs, which are manufactured or man-made. Now, these can, when we look at these, we want these drugs or these chemicals to be very selective in order for them to be effective. All right, we need them to selectively fight what we need it to fight to be effective and not cause a bunch of side effects to us. And so some examples of some key players in this, Ehrlich in 1910s, he came up with salvarsan. He thought it was the magic bullet to treat syphilis. We see that it's actually not, because it contains arsenic. So arsenic we now know is also harmful, so we don't utilize this in this way. But we do see that it was used to help treat syphilis. Fleming in the 1920s, he accidentally discovered penicillin. And this is a natural antibiotic created by a fungus that we can use. And there are different derivatives of penicillin now called amoxicillin, ampicillin, all these different types. But Fleming is the scientist who helped discover that in the 1920s. Now, a problem is with a lot of times our antibiotics is we are now seeing that there are things that are antibiotic resistant. These are strands that merge over time. as antibiotics are used and they're misused, like they're used too often, they're not completed by the patient self-medicates using the wrong kind of antibiotics. This all creates these types of microorganisms to mutate, and they become antibiotic resistant. And we'll talk more about that when we get to the chapter that talks about antibiotics and different medications that we use against microorganisms. So what are some fields of microbiology? Well, there are lots of different ones. These are all under the umbrella of microbiology, but you have bacteriology, which is the study of bacteria. You have mycology, which is the study of fungi, and then you have parasitology, which is the study of parasites. All right, these parasites are things like protozoas and helminths, okay, worms. All right, so there are some subgroups. So you could be a microbiologist who studies kind of all these, or you could specifically just study bacteria, or you could specifically just study parasites. Okay, some other fields that are related more to the medical side, which are not dealing so much with the organisms, but how our bodies respond to them, is something like immunology. This is the study of immunity. We also see the recombinant DNA technology, which is more of our gene mutation. Okay, how do we mutate it to help with different kinds of things with microbiology? And then you have virology, which is the study of viruses. All right, and so these are also going to be fields of microbiology. They just may look at things a little bit different. Okay. So microbes, how do they affect humans? Well, in reality, we do see that a lot of them are beneficial. They do have an ecological benefit. This is where they recycle vital elements within our ecosystems, like a lot of our chemicals. We see chinops here, which is carbon, nitrogen, oxygen, phosphorus, and sulfur. They help recycle a lot of those as decomposers. They are used in sewage treatment plants to reclaim water, to clean water. Actually, in our hometown, my hometown, we have this, where they use the microorganisms, and it also helps with cleaning the water and creating a more natural fertilizer. GMOs are used to clean up pollutants. So these organisms, these microorganisms, have been genetically modified in order to do something they normally don't do, like clean up oil spills or any kind of toxic waste that could be present. These can provide safer way to control agricultural insect pests in the sense of not using chemicals, like not spraying them with pesticides, but instead using microorganisms to help protect those plants. Some medical and biological benefits that we see that can be present as well are some of these guys do produce antibiotics. They can be used in gene therapy research. We see recombinant DNA can be used to make vaccines, insulin, growth hormone, and even enzymes that people are missing. They can generate crops that are hardier, like they're drought resistant. They can maybe these plants can grow in higher temperatures or lower temperatures, insect resistance. They can improve our food products by creating like super fruits and vegetables. We see that those are like the bigger strawberries, or they have a better shelf life. They last longer. We also see that on us, we have what we call our normal microbiota or our normal flora. These are microorganisms who live on our skin or in our body that help protect us. They can supply us with vitamins. They can protect us against other types of bacteria. They are actually helpful to us. And then how are they helpful economically? How do they help us money-wise? Well, they can produce products such as alcohol, acetone, and vinegar. We also see that some microbes can help us make food like sauerkraut, pickles, soy sauce, cheese, yogurt, bread. Microorganisms help us make those. They can be used in energy in industries that help create biodiesel or clothing products. So like if you ever have had stonewashed jeans or jeans that they'll talk about how they have like a special kind of treatment on them, they probably used microorganisms to help with that. Now, economic disadvantage though, these guys can cause food spoilage and water contamination, which is not good, and that can cost us money. But for the most part, we do see that there are some really good economic benefits. All right, so what about the pathogenic side? What about the ones who cause disease? Well, there is a group that's called the infectious diseases, and these are caused by bacteria, viruses, fungi, and parasites. They can be passed from person to person. Okay, like by respiratory droplets, like if I cough, it gets in the air and you were to breathe it in. This is what happens with diphtheria. They could be transmitted via insect bites or by bites from animals. Malaria is transmitted through mosquitoes. West Nile. Rabies is transmitted if you're bit by a dog who has rabies or another animal who has rabies. Acquired by environmental exposure. Sometimes it's due to you drinking contaminated water or eating contaminated food. We see this with cholera. So there's a lot of ways you can get these diseases, but they are infectious, meaning they infect your body. Some emerging infectious diseases, these are newer or changing diseases. So some of these are going to be like, wait a minute, the flu has been around for a long time. We've known about it a long time. However, it changes constantly. And so because of this, we need to study them more and really watch their incidence rates, like how often they occur. Because as they change, we need to try to stay ahead of them if at all possible. So viruses are things like SARS, H1N1 influenza, avian flu, West Nile, Ebola, HIV. Coronavirus would be part of this as well. Even though coronavirus is just a common cold, when we look at COVID is the coronavirus being mutated and it's very different. We also have purons. Purons are proteins that can change, and this is like Creutzfeldt-Jacobs disease or CJD. And then there are some bacteria that as well that have mutated and become more antibiotic resistant, which makes it harder to treat. This is like MRSA or VRSA, C. diff, and E. coli. All right, and so these are just some examples. These are not the only emerging infectious diseases. There's a lot of them, but these are just some examples. So when we look at this, based on what you've learned so far, are the majority of microorganisms beneficial or pathogenic? Well, hopefully after this, you're answering that they are mostly beneficial. This is because most of them are helpful to us or they don't harm us. They may help our ecosystem, but they don't maybe help us directly, but they're not hurting us. Okay, but again, the ones we tend to focus on and the ones we tend to think are cool to learn about are the pathogenic ones, which in reality is what we're going to spend most of our time looking at. But in reality, most of them are beneficial. All right, so as we dive into these different topics about microorganisms, please feel free to ask any questions that you may have.